Hello everyone, welcome to my channel. So, I encourage you to subscribe to stay informed on the latest topics and we delve into the captivating subjects with clarity and insight. Thank you for joining this journey. I look forward to sharing more engaging content with you. So, today we are diving into the world of pharma chemistry, focusing in the mystery in analytical chemistry as it relates to the ICH-Q1 stability guidelines. ICH plays a crucial role in setting global standards that ensure drug quality and safety. Our goal today is to unravel how these guidelines help maintain the stability and effectiveness of pharmaceuticals. We will explore the study guidelines that form the backbone of drug development, ensuring that every medication meets rigorous safety and quality standards. In this overview, remember that these guidelines are not just about stability, they are about fostering trust in the medications we all rely on. So let's get ready to see how these standards unify practices across major agencies like the FDA and EMA, ensuring consistent drug safety worldwide. Imagine this: a medicine sitting on a shelf looks like perfectly fine, but inside it's slowly degrading. That's exactly why stability testing exists. In the world of pharmaceuticals, following, following the International Council for Harmonization guidelines is not just a requirement. It's what ensures every drug remains safe, effective, and high quality until the very last day of its shelf life. So today I'm going to simplify the stability testing as per ICH Q1A from real-time studies to accelerated conditions and show you why even a small change in temperature or humidity can impact your product. So we will go to the overviews. So ICH Q1 guideline is crucial because it provides a harmonized approach that spans the FDA, EMA, and PMDA, ensuring consistent evaluation of drug stability worldwide. This consistency is vital as it guarantees that drugs maintain their safety, efficacy, and quality throughout their shelf life. So let's consider why this matters. Firstly, these standards form the backbone for regulatory submissions across different regions, facilitating a smoother process for pharmaceutical companies. Additionally, they establish the scientific basis for determining the expiration dates and storage conditions, which are critical for both manufacturers and consumers. By adhering to these guidelines, we achieve three key objectives: global harmonization, quality assurance, and regulatory compliance together. These aspects ensure that patients can trust the medications they rely on every day. As we move forward, we will explore the comprehensive framework of the ICH Q1 series and how it further supports global regulatory harmonizations. So, today we are diving deeper into the ICH Q1 family of guidelines. These guidelines are crucial in establishing the robust framework for pharmaceutical stability testing, ensuring the global regulatory standards are harmonized effectively. The ICH Q1 series is divided into the four key guidelines. There is one more I will explain in the later. So ICH Q1A, this is the core guideline that outlines the stability testing requirements for new drug substances and products. It is the foundation upon which all other guidelines are built. So second one is ICH Q1B. This guideline sets forth the requirements for photostability testing. It's particularly important for products that are sensitive to light, ensuring they remain stable when exposed to light over time. So ICH-Q1C it focuses on stability testing for new dosage forms that are developed after the initial approval of a drug. This ensures that any new forms maintain the same high standards of stability and quality. So ICH-Q1D, this guideline is about optimizing the testing process. It suggests strategies like bracketing and matrixing designs, which help in the reducing the testing burden without compromising on the integrity and reliability of the results. So by understanding these guidelines, we can ensure that all pharmaceutical products remain safe, effective, and of high quality throughout their shelf life, regardless of where they are manufactured or sold. Okay, so what is the purpose of the stability studies? So the stability studies plays a crucial role in the pharmaceutical industry. First, these studies are essential because they help us to determine how long a drug can remain safe and effective for patient use. This is vital for patient safety and ensuring that medications maintain their intended quality throughout their life cycle. Next is stability studies help us to identify the optimal conditions for storing the drug by understanding the right temperature, humidity, and light exposure. We can ensure the product's integrity is preserved from the manufacturer to the patients. Ultimately, the goal is to establish a reliable shelf life and expiry date, which are informed by the ICH-Q1 guidelines. These guidelines ensure that the drugs we rely on every day are safe and effective until very last dose. Remember, our primary focus is always on protecting the patients. Okay. So, as we move into the discussing storage conditions, it's important to understand that these conditions are critical for ensuring the stability and quality of pharmaceutical products. The ICH-Q1A guidelines specifies three standard storage conditions: long-term, intermediate, and accelerated. These conditions are designed to simulate various environmental scenarios and climate zones, ensuring that the drug products perform well across different settings. So let's break these down. So one by one, that is the long-term conditions are set at 25 degrees Celsius with a relative humidity of 60%. This scenario is crucial for understanding how a drug performs under normal storage conditions over a time. So next one is the intermediate conditions involve a slightly warmer environment at 30 degrees Celsius and 65% humidity. This helps test the drug stability in slightly more stressful conditions, often encountered in the certain geographic regions. The next one is the accelerated conditions, that is at 40 degrees Celsius and 75% humidity, are used to predict a product's shelf life by simulating storage in a high temperature environment. This is particularly useful to identify any potential degradation that might occur quicker than expected. So each of these conditions provides valuable insights into the how the drug will hold up under different circumstances, aiding in the determination of a safe and effective shelf life. So understanding these conditions sets the stage for our next discussion on study timeline and testing intervals. Let's see how these conditions translate into real world testing scenarios. So, when discussing the stability timeline for stability testing, it's crucial to understand that establishing a comprehensive schedule is key to ensuring drug safety and efficacy over a time. So, according to the ICH Q1A guidelines, we need to follow specific testing intervals. Let's start with the first year. The tests are conducted initially and then at third month, sixth month, and ninth month and 12th months. So afterward, testing continues every 6 months up to 24 months and then annually. So this approach provides a thorough assessment of the drug stability at the different points in its shelf life. The initial test serves as the baseline, providing our first stability checkpoint. At the accelerated study endpoint, we focus on identifying any changes that might occur under stress conditions. So, long term validation, on the other hand, ensures that the drug remains stable right through the end of its shelf life. So, by adhering to these timelines, we can confidently determine the full shelf life of the drug, ensuring it remains safe and effective for patient use throughout its life cycle. Okay, so when discussing the testing parameters outlined in the ICH Q1 guidelines, we are focusing on the critical aspects that help ensure a drug's quality, safety, and efficacy over time. First, let's talk about physical appearance. This includes the checking the color, clarity, odor, and form of the drug, which are all essential for the maintaining the consumer confidence and ensuring there are no visible signs of degradation. Next, we have the chemical assay, which involves the testing the potency of the active ingredients. This is crucial because it ensures the drug remains effective throughout its shelf life. We also look at degradation products to identify impurities and set limits for them. This helps in understanding how the product might change over time and ensures that it remains safe for consumption. So, lastly, the dissolution rate is evaluated to understand the drug release profile. This is particularly important for ensuring the drug delivers its therapeutic effect consistently. So, these comprehensive tests conducted at regular intervals provide vital data, allowing us to establish the appropriate shelf life and storage recommendations for pharmaceutical products. So, the next one is the batch selection. So, when we talk about the batch selection in the context of stability testing, it's crucial to understand why this step is so important. So, according to ICH guideline, the batches we choose must be representative of the product that will be marketed. This means using the same formulation and manufacturing process to ensure the data we collect is meaningful and can be relied upon for regulatory submissions. Remember, a minimum of 3 batches is required. This approach helps ensure that the data is robust and comprehensive. The batches should be at a representative manufacturing scale, closely mirroring what will actually be produced for consumers. Additionally, using the primary packaging that is identical to what will be used on the market is essential. This ensures that the stability data accurately reflects how the product will behave in real world conditions. So, these steps collectively help us to establish a reliable shelf life and ensure the safety and efficacy of the drug product. So, the next one is the photo stability testing. So, photo stability testing, as outlined in the ICH Q1B, is crucial for understanding how light exposure affects the stability of pharmaceutical products. This guideline helps ensure that products maintain their quality even when exposed to light during storage and use. So, let's delve into some key aspects. Firstly, photo stability testing involves a minimum light exposure of 1.2 million lux hours. So, this rigorous standard helps simulate real world conditions where products might encounter varying light intensities. The forced degradation studies are conducted to identify potential degradation pathways. So, these studies are essential in understanding how light can alter a drug's composition and effectiveness. So, the lastly is confirmatory studies are required on both the drug substance and the finished product. So, it's imperative to evaluate protective packaging, especially for light sensitive drugs, to ensure they are shielded from illumination that could compromise their integrity. So, by understanding the photostability of a product, we can make informed decisions about its packaging and storage, ultimately, the safeguarding its quality and efficacy for consumers. Okay, so why the stability zones are very important here? So, we are tackling the classification of climatic zones. So according to ICH, which is crucial for determining the appropriate stability testing conditions for pharmaceuticals. Let's start with zone 1 and 2. So these are temperature zones. So which include regions like Europe, the USA and Japan. Here the standard storage conditions are between 21 degree to 25 degree with 60% of relative humidity. So these zones represent moderate climates where typical storage conditions apply. So moving on to the zone 3, which represents hot and dry regions. In these areas, the required testing conditions are 30 degree with 35% of relative humidity. So this ensures that products remain stable under drier conditions. So lastly, we have the tropical regions classified as zone 4A and zone 4B. So these zones require testing at 30 degrees Celsius but with higher humidity levels of 65% RH for zone 4A and 75% RH for zone 4B. So this classification accounts for the challenges of maintaining drug stability in hot and humid environments. Okay, so initially I told you that the ICH Q1E guideline. So initially I explained only the Q1A, B, C, D. So what is Q1E? So when evaluating the stability data, it's essential to understand the ICH Q1E guidelines, that is data evaluation, which help us to establish the shelf life and support regulatory submissions. First, we will focus on the statistical analysis. So, by using the regression, we can determine the degradation rates of the drug product over time. So, this approach allows us to predict how long the product will remain stable under specific conditions. So, another crucial point is the concept of extrapolation. That is, ICH-Q1E permits extrapolation up to two times the period of accelerated data. This means we can forecast stability over a longer time than initially tested. So, provided the data supports it. Finally, determining the shelf life at a 95% confidence interval ensures the proposed shelf life is statistically robust. We also assess data pooling for batches that show similar degradation patterns, which can streamline our evaluation process. So remember, the goal is to make confident data driven decisions that support the safety and efficacy of the drug product throughout its shelf life. So the next one is stability report documentation. So it's crucial and importance of thorough documentation in the construct of ICH guidelines. So the comprehensive stability reports are a cornerstone of regulatory submissions, serving as vital proof of compliance. So these reports must include several key elements. One is the stability protocol, which highlights the importance of detailing the studies, objectives, design, and acceptance criteria. This sets the foundation for the entire study and ensures that it aligns with regulatory expectations. Next one is the validated analytical methods. So this is the necessary of what is the necessary of using the validated methods and specifications. So validated methods and specifications demonstrate the reliability and accuracy in the data collected. So third one is the data tables and analysis. So complete data table should be provided showcasing statistical trend analysis. This not only displays the robustness of the study but also supports the conclusions drawn. So last one is the clear conclusions. Convincingly supports the proposed shelf life and storage conditions, providing a clear rationale for regulatory authorities to approve. So well organized reports facilitates a smoother regulatory review process and are essential for achieving global market authorization. Okay, so what are the common challenges we face during the stability studies? In our work with ICH Q1 stability studies, we often encounter several common challenges. These challenges require careful planning and robust protocols to ensure both regulatory compliance and data integrity. First, let's address the out of specification results. So, if the results are OOS, so these require thorough investigation to understand the root cause and implement corrective actions. It's crucial to remember that each investigation strengthens our process and compliance. Next is the identifying the degradation pathway is essential. So this involves understanding how and why a product might degrade over time. So by doing this, we can better predict shelf life and ensure the product efficacy. So another key challenge is container closure integrity issues. So ensuring that packaging adequately protects the product is vital for maintaining the stability throughout its shelf life. Lastly, the impact of climate zone, that is the variations cannot be underestimated. So different regions can have varying effects on product stability, necessitating the tailored stability studies to account for these differences. So by being aware of these challenges and addressing them proactively, we can enhance the reliability of our stability data and support successful global product registrations. Okay, so what are the best practices we can do during the stability studies? So we are delving into the best practices which are essential for ensuring that our stability studies not only meet the regulatory compliance but also support successful product registration across global markets. So first, it's crucial to define clear protocols. So these should include the justified storage conditions and intervals tailored to your product. This clarity helps to prevent any ambiguity during regulatory reviews. Next is maintaining the comprehensive records is vital. This includes all stability data and any deviations encountered during your studies. Such thorough documentation can be a lifesaver when questions arise during the approval process. Additionally, implementing ongoing stability programs for marketed products helps in adapting to any unforeseen changes and ensures continual compliance. Lastly, make sure to use the validated analytical methods. These methods should have documented accuracy and precision to ensure the reliability of your data. So, by adhering to these best practices, you are setting your product up for a smoother journey through regulatory submissions and approvals. So, paving the way for market success. So, as we wrap up, I want to express my heartfelt thanks to each of you for your attention. So, we have covered a lot about the ICHQ1 guidelines, from challenges to best practices that ensure compliance and integrity in stability studies. So, your engagement and curiosity throughout the session have been invaluable. If you have any questions or comments, please feel free to drop them in the comment section. For further queries or to continue this conversation, you can reach me at my mail ID. It's in the slide. So don't forget to subscribe to our updates for more insights and upcoming sessions. So thank you once again for being here, and I look forward to our continued discussions. Thank you. Bye bye. Take care. Enjoy your day. Bye.